Welcome to this pain clip on our recent publication about dorsal horn GBX1 neurons and their role in stress-induced analgesia. My name is Uli Zeilhofer, and I'm a professor of pharmacology at the University of Zurich and at the Swiss Federal Institute of Technology.
Hello, everybody. My name is Robert Ganley, and I was one of the authors involved in this study.
As we all know, the intensity of a painful stimulus we feel is not only determined by the peripheral nociceptive input, but is strongly context dependent. It can be internal states, it can be environmental factors. And one very important environmental factor which can induce strong analgesia is acute stress exposure. We all know this from victims of traffic accidents or sports athletes during competition. Even in the presence of severe injury, they sometimes don't feel the pain at all. Now, why GBX1 neurons? Our group has a long-standing interest in inhibitory dorsal horn neuron populations. And in a previous study, we had looked at glycinergic neurons, which are inhibitory dorsal horn neurons in the deep dorsal horn. And when we interfered with their function, we found strong phenotypes in nociceptive tests. But we were also interested in the inhibitory neurons of the superficial dorsal horn. Many of them are purely GABAergic. And Hendrik Wildner, a senior scientist in our group, came across an interesting transcription factor, gastrulation, brain homeobox factor 1, which is expressed in GABAergic neurons of the dorsal horn between lamina 1 and lamina 3. And Karin Hanlitz, a previous PhD student in our group, she generated a GBX1 Cree mouse to functionally interfere with this interneuron population.
So the first thing that we wanted to do with this new GBX1 Cree animal was to use it to functionally study these neurons during behavior. So the first experiments that we decided to do was to chemogenetically inhibit these neurons using HM4DI and CNO injection. What we found was during this chemogenetic inhibition, there wasn't much of a difference in the responses to different types of peripheral stimulation. So, for example, the responses to heat, mechanical, and cold wasn't affected at all by this chemogenetic inhibition. The next thing that we decided to do was to see what these neurons had the capacity to do. So when we chemogenetically activated these neurons using HM3DQ, the neurons were able to very strongly suppress behavioral responses to heat, cold, and mechanical stimulation. So this made us think what naturally occurring conditions exist in which these neurons would normally be silent or less active, and then suddenly very strongly activated. And this is why our thoughts turn to stress-induced analgesia, because in these conditions, acute stress will cause a very strong activation of some neurons, which in turn will or should inhibit sensitivity to nociceptive stimulation. So we used a model of stress-induced analgesia called swim stress-induced analgesia. So because mice don't enjoy swimming, they're not waterborne animals, they become very stressed after very brief exposure to water. And this produces a very stereotyped time-dependent increase in the withdrawal latencies and withdrawal thresholds. When we chemogenetically inhibit the GBX1 Cree neurons, this increase in the withdrawal latencies and withdrawal thresholds is almost completely gone. So when we chemogenetically silence the GBX1 neurons, this stress-induced analgesia is effectively abolished, which suggests that these neurons are a critical component in swim stress-induced analgesia.
Now, this was a very nice phenotype, but we were interested in the underlying neuronal circuit. And we used optogenetics-assisted circuit tracing to find out how these GBX1 neurons inhibit pain. And we had two hypotheses. The GBX1 neurons might either inhibit output neurons, nociceptive output neurons, via classical postsynaptic inhibition, or they might reduce nociceptive input to the spinal cord via presynaptic inhibition. And what we found was activation of the GBX1 neurons had a strong effect on the nociceptive output neurons, but little or no effect on nociceptive input to the spinal cord.
So, of course, stress is perceived and experienced within the brain. And from there, it's signaled and activates all of these stress-activated pathways. So we hypothesize there must be some sort of descending connection between the brain and the GBX1 neurons in the spinal cord. So in order to study the direct descending inputs to these neurons, we employed monosynaptic rabies tracing as well as optogenetic-assisted circuit tracing. So the first experiments we did was to perform monosynaptic rabies tracing from the GBX1 starter population and see which brain areas provide direct input to these cells. Although we found many areas providing direct input to the GBX1 neurons, we found the vast majority of them were located in the RVM in the hindbrain, which is an area which is already known to play an important role in descending pain modulation. When we looked at these neurons in more detail, we found that most of them expressed markers of inhibitory neurons, suggesting that the main input to these neurons from the brain is a descending inhibitory pathway that is found in the RVM. We also confirmed this using optogenetics. So by optogenetically activating the descending pathways from the RVM to the spinal cord, we were able to measure exclusively inhibitory currents in the GBX1 neurons. When we also traced the excitatory and inhibitory descending pathways from the RVM to the spinal cord, we found that only the inhibitory pathways were able to innervate the area where the GBX1 neurons were found, suggesting that this is the pathway which controls GBX1 neurons in the level of the spinal cord. So what we would predict from this is, since these neurons are mostly receiving inhibitory input from the RVM, in order for them to be activated by this pathway, we would hypothesize a disinhibitory circuit in which there's a descending inhibitory input to the GBX1 neurons, which is normally active, but is removed during acute stress, which then allows these neurons to become more active and mediate stress-induced analgesia.
The previous work has shown that serotonin also plays a major role in stress-induced analgesia. So we were asking, is there a role for serotonin in the circuit we have characterized? And it is well known that during acute stress exposure, there's a lot of serotonergic input from the RVM to the dorsal horn. So the first hypothesis we tested was whether serotonin would interfere with the biophysical properties or the firing patterns of GBX1 neurons, but it did not. So we asked another question and tested whether serotonin released in the spinal cord might interfere with the release of GABA or glycine from the descending inhibitory RVM neurons. And indeed, we found that this inhibition was about reduced by 50% by serotonin. So serotonin release might be another way by disinhibition of this stress-induced analgesia circuit. So if you find this study interesting, please feel free and read the full story in Neuron.